So we are very happy to have Dr. Adanaska Marinova-Petkova from the CDC joining us. She is the lead of the National TB Reference Laboratory at the CDC's Division of TB Elimination and oversees the Molecular Detection of Drug Resistance, or MDDR service. She's board certified. She's a board certified clinical and public health microbiologist and serves as the CLIA technical supervisor and clinical consultant for the reference laboratory. She holds a Doctor of Veterinary Medicine, a Master's of Molecular Biology and Biotechnologies, and a PhD in Epizoothology and Infectious Disease and Animal Degrees. She's been at the TB Reference Laboratory since 2019 and has been the lead since 2021. We are very happy to have her, and I'm sorry for going over with our case. Please take it away, Adanaska.
Thank you. Hi, everyone, and I'm really happy to be here today. Let me share my screen. So I am going to talk about Mycobacterium tuberculosis susceptibility testing for pyrazinamide, or PZA. And I was supposed to give this lecture today with my colleague, Glenn Morlock, who is a microbiologist with our branch for 32 years, but he had an emergency and he couldn't be here today. So bear with me, the first half of the slides are his, but I will do my best to present the science behind the PZA test. So PZA is a pretty old drug. First clinical trials with PZA have been published in the 1950s. And today, this drug is part of the first-line regimen for pulmonary drug-susceptible TB. It has bactericidal effect against Mycobacterium tuberculosis and is more active against the older, slowly replicating or non-replicating bacilli, and less active against the young, rapidly replicating cells. The drug is also active within the macrophages. It allows for shortened TB regimens, and it has synergistic effect with other anti-TB drugs, like from the older groups, rifampin and moxifloxacin, for example, and from the newer and repurposed drugs, bedaquiline, delamanid, and pretomanid. A little bit about the PZA in vitro activity. So PZA is analog of vitamin B3 or nicotinamide. And it's in fact a prodrug, which means that once in the organism, it gets converted into pyrazinoic acid and ammonia as a byproduct by the enzyme pyrazinamidase in a chemical reaction that is called deamination that you see at the bottom of my slide. The enzyme that performs this function, the PZase, is encoded in the MTBC genome by the PNCA, or we commonly call this PNK gene. And please note the ammonia, once again, which is a byproduct, because this is a troublemaker and will come up in my talk in just a minute. Some factors that affect the PZA activity, and the number one is the pH of the medium. So as you can see, the PZA requires a pretty narrow pH range. About 5.6 to 6.0 is the best range for the drug to be active. When the media becomes too alkaline, there's no PZA activity. And when it's too acidic, there's inhibition of growth in the drug-free media. So basically, theoretically, based on the Henderson-Hasselbalch equation, with every increase of pH by 0.3, the PZA MIC doubles. So the bottom line is, as the media neutralizes, PZA becomes less active, and therefore the MIC increases because it requires more PZA drug to inhibit the growth of the microorganism. Another factor that is affecting the PZA activity is the inoculum size. So a large inoculum reduces the PZA activity, and this is much more pronounced with PZA than with other drugs. The commonly used inoculum that we use in our test has approximately 10 to the power of 6 cells in 1 ml. And that already increases the pH of the media with 0.3 units. If you go from 10 to the 6 to 10 to the 7 cells in ml, the PZA will become completely inactive because at 10.7, the pH already increases by 1 unit, and at 10.8 by 1.5 units respectively. So the culprit here is the ammonia that is produced by the process of deamination of the PZA and causes the pH to increase. Another factor that affects PZA activity is the bovine serum albumin, or BSA, which is a common media supplement. In the MGIT test that I'm going to look at in a minute, it is at 5.0%. I'm not going to go into too many details, but basically, high BSA concentration can neutralize the media as well. Because if it increases between 5 and 10% in the medium, the pH is going to increase by 1 unit. So let's look at the phenotypic methods for PZA drug susceptibility testing and some of the challenges. There are a couple of types of methods for phenotypic detection of PZA. The indirect method detects PZAs, or pyrazinamidase activity. And it's also referred to as the Wayne test based on the name of the developer. This is a colorimetric test, and when the tube, like old-fashioned test, when the tube turns red, this means that pyrazinamidase is positive, which means the isolate is PZA susceptible. And if there's no color, then the PZase is negative, or the PZA is resistant. So the problem with this test is that it is indirect. The resistant is diagnosed by a negative result. And versus all other susceptibility tests, resistant is diagnosed by a positive result, basically growth in the presence of drug. And logistically, the test is not very convenient. It's slow, it's laborious. There are some safety concerns, and it's difficult to QC. I've personally never done this test or seen it, in the lab. I've just read about it. We don't use it in our lab. And as part of direct methods, these are the culture methods for detection of PZA resistance. The growth of the microorganism is detected in the media or on the media that contains the drug. They're also problematic for PZA because both resistant and susceptible bacilli can be inhibited by the very acidic pH and is affected by the inoculum size. Also known to some of these culture methods to have poor reproducibility. And there's been quite a lot of false resistance detected and published in various studies through the years. The PZA false resistance has become increasingly common with the transition from the BACTEC 460 method to the MGIT method that we are using today. So the BACTEC 460 method is a radiometric, or was a radiometric method that measures C14 labeled carbon dioxide in the bottle head space of the vials. And it uses less enriched media with less supplement, which is at only 0.5% BSA. And the reason this test got discontinued in August of 2012 was the safety, because it was radiometric using labeled isotopes, as well as it was using sharp needles. The method that we are currently using, the MGIT, or the Mycobacterium Growth Indicator Tube method, uses fluorometric detection, which increases with the depletion of oxygen from the bacterial respiration in the tube. And it has more enriched media. The BSA in this media is at 5.0%, so 10 times more enriched, which is for the purpose of promoting bacterial growth. I'm sharing here a slide from the MPEP challenge. So MPEP stands for Model Performance Evaluation Program, which is administered by the CDC. Oops, I'm sorry, that was too fast. And this program is a voluntary program for laboratories across the United States that perform drug susceptibility testing. And it serves as basically alternative proficiency testing challenge instead of official PT. So twice a year, we ship 5 isolates to various laboratories, public health, government, private sector labs. And in this particular challenge, this is from November 2010. We shipped 5, this is prior to my time, of course, when I say we, but 5 samples were shipped, but they were representing 3 strains because there were 2 that were duplicates. So as you can see here, 17 laboratories performed BACTEC 460 method for the 5 isolates, and they did not have any problems detecting them as susceptible. By the way, these were selected to be PNK wild type isolates. But as you can see from the laboratories that use MJIT for the detection of PZA, for isolates C and D, about 30% of the labs got these 2 isolates as false resistant instead of susceptible. So this pretty well kind of summarizes and illustrates the difference between the 2 methods and the problem. Just in a couple of words, the methodology, the principle of the MJIT tube, it contains a fluorescent compound embedded on the bottom of the tube, and which contains fluorophore, and the fluorophore is quenched by the oxygen at the beginning of the test. But as the bacteria grow and breathe basically inside, they deplete the oxygen, and then the fluorophore starts fluorescing, and the machine detector is detecting this at a certain intervals and flags the tube as positive. And this is basically a schematic of how the test works. You would have 2 vials of the same isolate. One would be at 10-fold inoculum dilution and no PZA drug in it, and this is going to serve as a control for the test. And the other one will have PZA drug at the critical concentration, which is 100 micrograms per ml. And then both are placed at 37 degrees in the instrument. And the control, as long as the control grows up to 400 growth units between day 4 and day 21, the test is considered to be valid. So at the time when the control reaches 400 growth units, the test will stop, and we'll evaluate the tube that contains the PZA drug. So whenever the growth units are 100 or above, the test is considered positive or resistant to PZA. And whenever the growth units are below 100, the test is considered negative, or the isolate is considered as being PZA susceptible. This test tends to be very temperamental. If the inoculum density is too low, the test will terminate prior to the result. But if it's too large or heavy, the media is going to be neutralized. So there will be false resistance. And the inoculum must be just right for the test to be accurate, but that's, that might be problematic to achieve sometimes. And from studies that have been done, it seems like the devil is in the seed tube, because for you to get to the PZA actual testing, you need to first grow this in a MJIT seed tube. And so the test allows you to use this seed tube once flagged positive for growth within 5 days. From day 1 and 2, you use it directly without dilutions, but days 3, 4, 5, you need to do for the test insert, the package insert, you need to do dilution to use it. But considering that the test tube may become positive at various times during the day, this can be very challenging. I'm not going to go into too many details here because this is, a little bit of, lab specific. But basically, there are challenges with it. So the question is this BD protocol for MJIT PZA too imprecise? with the too large time of a time window for 5 days where you can use this tube, and how can this be better, measured? Perhaps smaller time windows of when the seed tube becomes positive, but this may be constrained by the work process because the labs don't tend to work 24/7 and set up PZAs at any time of the day. And then the growth units and various isolates may grow at a different with different speed. So again, the test is challenging. There have been efforts through the years to address the problem with this test. I'm showing here a study that was published. And the first author is Glenn Morlock, who was supposed to be here today. This is one of the reasons why I invited him to be my co-speaker. So basically in this study, just briefly, 9 laboratories participated, and they tested 3 inoculum. The BD standard inoculum per the package insert, as well as method A, where they diluted the inoculum 2.5 times, and method B, where they diluted the inoculum 5 times. And the false resistant results declined nearly 4-fold from 21% to 5.7% using method A, so 2.5 times diluted inoculum. And the positive predictive value of a resistant result was greatly improved using the lower density inoculum. The method A inoculum time for the test completion increased by 1 day, from 9 to 10 days, based on this study. So some considerations when you are dealing with mono, PZA resistant results, because we mentioned that there is, this tends to be some false resistance. So the first question that we all need to ask is this M. bovis? And if it is, that it's normal to be PZA resistant because it has inherent resistance. But then if it's not M. bovis or there's no indication for that, another thing that could be done is to consider molecular testing for the PNCA gene. However, there are very, small number of isolates that are mono-PZA resistant with PNK mutation. Usually these mutations are found in the MDR-TB isolates, or where there is more than one drug resistant. So you should be suspicious for false resistance in those cases. So if false resistance is considered, the lab should also consider what is the possibility of over-inoculation in the test. And there are a couple of pathways here. To stick with the BD MJET like package insert with the test, which is a FDA cleared test, perhaps repeat the test and use, younger seed tube day 1 versus day 2, or day 3 versus day 4 and 5. For the last 2, you need to, for the package insert, there is dilution that needs to be made before you add it to the test. Record hours, and then avoid clumps. This is something that we've been practicing in our lab. Allow some time for the seed tube after it's been well vortex to settle down, and then aspirate only from the top of the vial to avoid over inoculation. But then some labs have decided and may decide to take a different route and to pursue what was published in that study and use some kind of reduced inoculum perhaps. But you should always remember that once this anything is done differently than the package insert, this test is no longer considered FDA cleared test. So before any of this is done, full validation is required, and the test needs to be approved by the CLIA laboratory director. So how do we do PZA at the CDC? Basically, we use the package insert. We use the test as a FDA clear test, and we do not use the reduced inoculum in our laboratory, but we do make sure that there are no clumps, and we have very low turnover of staff. So the people who work here in our lab tend to work for many years, and they do understand well and perform the same way the test. Additionally, we perform complementary PNK sequencing and reporting. This has been done for many years in our lab as part of the MGDR service, but a couple of years ago when I came on board, I made this mandatory for every test that is for PZA susceptibility test order or for molecular identification, which is a specific test for M. bovis and BCG differentiation from the rest of the complex, to have PNK sequenced and reported with it. However, the lack of PNK mutation does not necessarily rule out phenotypic resistance to PZA. How do we report our results? So if we get a PZA susceptible result, because we have never had an issue or we haven't found any studies published with a problem of false susceptibility of the test, we may not even perform PNK sequencing, and we will report this as susceptible without any repeat testing. Whenever we have a PZA resistant result, by MGIT, if there is a PNK mutation, we necessarily do that. If there is a PNK mutation that confers resistance or likely resistance or has effect unknown, we are going to report this as PZA resistant result and not necessarily repeat the test. However, whenever we have a resistant result by MGIT, without PNK mutation detected or wild type sequencing, we repeat the test. If on the repeat, we again have MGIT PZA resistant result, we are going to report this as resistant. And if we receive, because there are cases when you would have on a repeat susceptible result, we report this as unable to get reliable results. And we put an additional comment why, because on the test, it tested as resistant, but on repeat, it was susceptible for the clinicians to know what exactly happened here. The majority of true PZA resistance is due to mutations in this PNK gene. It encodes the pyrazinamidase that I talked about at the beginning of my presentation that converts the PZA to POA. And M. bovis is an example that is inherently PZA resistant due to this His57 mutation in PNK. There is unfortunately, a hotspot in PNK for mutations, unlike rifampin, where we have the RRDR, where we know the mutations are mostly concentrated there, or like the QRDR for fluoroquinolones. There's no such thing with PNK. Mutations occur anywhere in the promoter or the entire coding region of the gene. So the effect also of many of the PNK mutations that are detected on the PZA resistance phenotypically is unknown, because there is this lack of reliable phenotypic test that makes it so difficult to determine the significance of this, of some of the mutations. There are other genes that have been published in the literature, such as CLPC1, RPS8, PAN-D, that play perhaps minor role in the PZA resistance. But even when you have labs that they perform whole genome sequencing, the interpretation still is typically only considers the mutations in the PNK. On this graph, I'm showing basically the PNK length of the region that we examine. That includes a promoter of -30 to 1, and then you have 187 codons in the gene. And these are all basically that you see on the graph are mutations detected at certain positions. And having in mind that the ASP57, His57 ASP mutation for bovis have been removed from this. So as you can see, mutations are found all over the gene. Some known problems with the MGIT PZA testing that affect the results is basically discordance is observed even in our lab here as a reference laboratory. But the thing is that the majority of the isolates that come to us for PZA susceptibility at CDC are already problematic or PZA monoresistant in the public health labs. So it's really difficult to assess, for example, if, if we're seeing too much PZA monoresistance, because these are already problematic. It's kind of a biased sample set that we work with. We do see quite a few PZA resistant isolates by midget that are after that susceptible, and they don't have PNK mutation detected. And we also see a fair share of PZA resistant twice that don't have PNK mutation, and they're not part of MDR-TB isolates. And there is another culprit to this, which is called lineage 1, or the Indo-Oceanic lineage, that is known to be associated with discordant results, and perhaps can explain a lot of this discordance. Often isolates from this lineage test as PZA monoresistant without having PNK mutations detected. These isolates have MIC for pyrazinamide that is close to the critical concentration, and that causes, with repeat testing, inconsistent results that could be resistant and then susceptible and flip back and forth. There is a published study that suggests potential link between a mutation in this CLPC1 gene in low-level PZA resistant in lineage 1, but there's more data is needed for this to be concerned. There's not enough published data on this lineage 1 effect. And these are basically the main reason for the low sensitivity of PNK sequencing assay when we compared it to phenotypic results when we went live with the targeted next generation sequencing for the MDDR service a year or so ago. The sensitivity of PNK was 69.8% for MDDR. It is, however, not known at this time whether this elevated MIC for lineage 1 has any clinical significance for the patient. Additional problems with the Amgen PZA testing. Last year in 2023, in February, March, our laboratory experienced a series of QC failures of the H37RB control, which is supposed to test as susceptible, and we started getting resistant QC. And there was a variability within single PZA drug lot for the test kit. So we were having both QC failures and QC passed with the exact same lot of reagents used. We did file complaints with Becton Dickinson, followed up with them, returned a couple of PZA drug lots to them to investigate, but they told us at the end that they didn't find any problem with this lot, so they didn't know what led to this. And for our lab, this problem led to unprecedented number of test repeats and extended turnaround times for the patient so that reports could be issued at all. In October of 2023, we started having the same, this first happened in February, March, and then we didn't have problems for a few months. And then in October, here we go again. It wasn't just us, but multiple laboratories throughout the United States, public health labs, commercial labs, reported QC failures and same problems. And some labs even reported elevated percentage of PZA resistant patient results, which we cannot assess in our lab because, again, we have a biased sample set, but in the public health labs, they usually know what's normal for their population. And they reported that as well to APHL and to BD. In December, January of this year, multiple labs in the US suspended the Amgen PZA tested, and we stepped in and performed testing for many laboratories that were experiencing problems by performing MDDR where applicable, by Amgen PZA testing, by PNK sequencing alone or in combination. And we also assisted APHL in discussions with BD to address this problem. So we combined the list of problematic reagents from 17 affected labs at the time. This list, I believe, grew after we started these meetings. And we also released a Dear Colleague letter to inform the public health labs of the current challenges. until today, the Becton Dickinson group is still working to identify what is the exact cause of the reported issues, and we're all waiting to hear more about what causes this and and how are they going to fix this, and is it fixed? So because there were a lot of questions about, okay, well, what do we do? Do we go to PK sequencing, or what is the solution? So I reached out to our colleagues, Jamie Posey and Warren Cohen from the applied research team, and requested some data based on the national surveillance data. This is when people that have TB, their first isolate is sent to Michigan for genotyping, and then the data are analyzed by CDC. So I asked them to, provide some data, some context of what is going on and what is the accuracy of, PK sequencing, et cetera. So based on the whole genome sequencing of culture confirmed cases of about 25,400 isolates that had whole genome sequencing data and pyrazinamide MGIT susceptibility data for the period between 2018 and 2022, they provided data after they applied the interpretation criteria of how the World Health Organization catalog of mutation identifies, basically interprets mutation, and our MDDR service as well. And they calculated the performance metrics and stratified the data by the major lineages, which is 1 through 4. And in this table, if you look at the numbers, so basically out of the 25,400 isolates that they looked at, about half of them fall under lineage 4, and about what is this, like 20% fall under lineage 1. If you look at from lineage 1, those 5,828 isolate, about 8.4% of this were mono-PZA resistant by the phenotypic method, by the MGIT PZA, versus all the other 3 lineages had less than 1% of mono-PZA resistant. And then if you look at the molecular data that correspond with this, out of this 492 isolates, 94.7% did not have a mutation predicting resistance. So molecularly susceptible, phenotypically resistant. U is for undetermined significance of detected mutations, which is about 1.9%, and then resistance was predicted molecularly for only like 3.5% of this lineage 1. If these are the metrics that they provided, sensitivity, specificity, positive and negative predictive values, and across the top is for all isolates that they looked at, like the 25,400 isolates, and then you have only MDR isolates considered, and then they're basically there is a breakdown by lineage. And again, if you look at sensitivity for MDR isolates, it's very high, as well as specificity. If you look at lineage 1, though, sensitivity is like only 5%. Specificity is very high. So these are, I guess, very, very useful data. And oops. I don't know why. I thought I had additional slides here, but not sure why I'm not. I hope I'm not missing something. So basically, if I have to summarize, this MGIT PZA's FDA cleared test, one of the few that we have in the field to use, but it can be very challenging with multiple issues that have been problematic since, the beginning of the usage of this test, like inoculum size, but there are also recent problems with the reagents. And lineage 1, which have elevated MIC close to the critical concentration, might be the reason for, or perhaps it is a reason for a lot of, post-resistance or discordance, I would call it. And whole genome sequencing accurately predicts drug susceptibility and resistance. And I already know of some laboratories that have dropped the MJIT PZA test and will only move forward with performing molecular testing for PZA. For our lab, we are currently analyzing data to make a decision. How do we want to move forward, especially if we don't get very soon, some kind of this problem with reagents solved. And then with that, I would like to acknowledge my team, the TB Reference Laboratory team, as well as Jamie and Lauren from the Applied Research team for providing data for this presentation. And Angela Starks, who is the branch chief, and Lois Diem, who's our quality manager. And with that, I am happy to take any questions. Thank you.